Good afternoon, everyone, to all of our attendees in the room and all of you online. Thank you for joining back with us for the afternoon. My name is Katharine Feibus, and I'm the team leader for the device evaluation team in the Division of Therapeutic Performance 1 in the Office of Research and Standards in FDA's Office of Generic Drugs. And I will be coordinating this session and will also be the second speaker. This session is called Other Design Differences, Justifying Differences in User Interface that May Affect an External Critical Design Attribute. Our first speaker this afternoon is Jason Flint. He's the Associate Director of Human Factors in the Division of Medication Error Prevention and Analysis 1 in the Office of Surveillance and Epidemiology. I'm going to let you read the sub-office name because it's very long. And I will be the second speaker. And then we will be joined by Claire McDiarmid, who is from Beatrice. She's the Senior Director of User Interface and Risk Management. Following the presentations, we will have a panel discussion and this is not working. Thank you. And we will be joined by additional panelists. Captain Irene Chan is a clinical pharmacist who is the Deputy Director in the Office of Medication Error Prevention and Risk Management in the Office of Surveillance and Epidemiology in CEDAR at the Food and Drug Administration. We will also be joined by Somesh Chattopadhyay, who is a team leader in the Division of Biometrics 8 in the Office of Biostatistics in CEDAR's Office of Translational Sciences. He is a biostatistician who works often with us on our comparative use human factors data, and will be able to share some of his statistical insights with us during our panel discussion. We will also be joined by Dr. Bill Chong, who is our Director of the Office of Safety and Clinical Evaluation in the Office of Generic Drugs. And Markham Luke will be making his return to the panel. He is our Director and Supervisory Physician in the Division of Therapeutic Performance. He is a dermatologist and has been in multiple leadership roles around the agency over the last 20 plus years. We are also honored to be joined by Nitesh Patel from Sun Pharmaceutical Industries, who is a senior manager there and will be joining us to provide some additional industry perspectives when it comes to evaluating and justifying other design differences. So I hope you enjoy the session. And Jason, come on up and take the helm.
So thanks for your attention. My name is Jason Flint. I'm an Associate Director for Human Factors in DMEPA 1. And this session or this talk is talking about best practices for comparative use human factors study design, execution, and reporting. Our standard disclaimer, the presentation reflects my views and should not be construed to represent FDA's views. And if we talk about anything that's marketed, any marketed products, that's just for illustrative purposes only. There's no endorsement there. So I wanted to start really from the beginning by describing the objectives of a comparative use human factors study. I want to go through the steps and the best practices for the design, and then talk about submission for CUHF protocols and study results and some best practices there. So previous presenters have talked about this, but this is the draft guidance for comparative analyses and comparative use human factors studies. And the focus here is on analyzing the proposed user interface relative to the reference listed drug or the RLD. And so remember that ANDAs rely on the finding of safety and effectiveness for the reference listed drug. This requires a demonstration of sameness of a number of characteristics, and those would be dosage form or route of administration or strength, and most of the labeling. And then generic combination products are classified as therapeutically equivalent to the reference, to the reference listed drug, and be expected to produce the same clinical effect and safety profile as the RLD under the conditions specified in the labeling. So, what does that mean? We're not establishing a new safety and efficacy for the proposed generic, and that's an important point. But we are trying to confirm that they're the same with the reference compared to the reference listed drug, and that implies a comparative approach. So previous subjects covered comparative analysis overview. I'm going to cover it very quickly here and then focus on one key point. This all starts with a comparative analysis. I'm looking for differences between the reference listed drug and the proposed product. And if there are no differences, right, then we're happy, right? And we don't need to have any other considerations. I would say that's rare. Many times there are minor differences. And if there are minor differences, these are differences that don't affect critical tasks, for example, then also this is okay. But sometimes there are other differences, other critical design differences. I don't know why that's advancing on without my guidance. Okay. But I think it's really important to note that if you do identify other design differences between your proposed product and your reference listed drug, that doesn't necessarily mean that that's going to end up with a comparative use human factors study. Okay. And so this is where you would want to have additional conversations with the agency and identify what additional information might be available, what other data might be available. Some of that data may come from a comparative use human factors study. Okay, so I just want to reiterate that. That's a sort of a key point. Just because there's another design difference does not mean that you necessarily have to proceed with a comparative use human factors study. Okay. So we'll get into the comparative use human factors study. Here, the objective is, so we've identified other design differences. We have determined that you need to perform a comparative use human factors study. And the objective of that study is to demonstrate that the differences that you have identified that are not minor wouldn't preclude approval of the proposed product in the ANDA. Generally, this is done through simulated use. And non-inferiority designs are generally appropriate. So the goal here is to show that the patient experience using a generic combination product without intervention, right, potentially from another care provider, will be no worse than that with the RLD. And there is some allowance for random variation. And we'll talk about that when we talk about D later. So when I'm trying to design this type of a study, I'm going to factor in all of these different things. The study design itself, what's sort of unique or what's happening with the user groups, what's the primary endpoint, justifying the non-inferiority margin, which then plays into justifying the sample size. And just to pause there, you want to consider any challenges that you might have or you foresee with recruitment, and then how you plan to address those. Randomization of subjects to different sequences, and then appropriate statistical methods for confidence interval and sample size. And my hats off to my colleague, Somesh, for those last set of slides that really came. I'm not a statistician, so Somesh keeps me out of trouble. So thanks. So for a COHF study design, typically this is a two-way crossover design. That's what we see the most often, assuming that there's no sequence, period, and carryover effects. And this is more efficient. This is chosen often because it's more efficient with respect to resources, with respect to the subjects, and to the number of products that need to be used in those studies. It's possible to do a parallel arm CUHF study, but we haven't seen that very often. In fact, I'm trying to think of one that we have seen. Typically, though, it's a crossover design. So when I say crossover design, this is what that means. Every subject gets both products. They get both the test product and the reference product. There's two possible sequences. They may get the test product first, they may get the reference product second, or vice versa. And then each of them are assigned to one of the sequences and then cross over. They're their own control, the subject. And typically this is a smaller sample size than if you were to do a parallel design. Okay, and so that's kind of drawn out here. I have a randomization scheme. My user goes to test first, the test product first, and then to the reference product, or to the reference product and then to the test product. So for user groups, the focus is going to be on whether substitution can occur with a full expectation that the generic product produces the same clinical effect and safety profile. So you want to include current users of the reference listed drug, and you should consider if your analyses indicate that specific subpopulation should be the focus of a study. And so you may see this in cases where I have multiple patient populations that are being treated by the same medication. So rheumatoid arthritis would be a good example. I want to focus on somewhat on the rheumatoid arthritis patients as opposed to something like colitis or something like that, right? I may expect that they have more difficulty, that population may have more difficulty using the product. And then finally, considering whether that difference in design may impact critical task performance for patients diagnosed with certain indications only. In terms of the primary endpoint, we've been recommending the overall use success rate. So although mathematically, this is just 1 minus the error rate, right? Success is the flip side of the error rate. So use success here is defined as not making a critical error, not making an error on a critical task by a subject for an application of a particular product. And usually only those critical tasks that may be impacted by the changes in the external critical design attributes are included in that primary endpoint. So again, endpoint considerations, primary endpoint is going to be the overall use success rate. The analysis is equivalent to that of overall use error rate, so you can use either. Ultimately, whether the delivery of the drug is successful is what matters. In some cases, depending on your product, you may have 2 or more co-primary endpoints that need to be looked at. So if you have different critical tasks for different doses, for example, you may need to look at that. So for non-inferiority, this is how we're testing for the overall use success rate. We're going to demonstrate that the overall use success rate for the generic product is not inferior to the RLD product for the critical tasks impacted by the change in those external critical design attributes. So there's a lot of talk about non-inferiority margin or delta. Should be determined on meta-analysis of overall use success rates for the reference product and similar patient populations, or formative studies or validation studies or preliminary studies can help you to get an estimate of that overall use success rate for the reference. And then some clinical judgment. So how much inferiority can you allow for that test device compared to the reference in terms of overall use success to not have sort of clinical impact? The key here is that non-inferiority margin must be pre-specified by the applicant, should be agreed upon by the FDA before the beginning of the study. I will say that this is an area of interest for the agency in terms of research and better methods to figure out what that non-inferiority margin is. And we have some broad agency announcements out now looking for some research on that matter. Some challenges that we see with non-inferiority margins that are submitted is justification. And so sometimes that margin is not justified appropriately. And then sometimes the applicant doesn't submit a protocol for review, submits the study report after conducting the study, and then it becomes difficult to know, okay, was the non-inferiority margin pre-specified or not? And then the challenge, of course, is that if that study is completed and then we don't agree with the non-inferiority margin or the justification, you may have to do another study. So again, the key here is we'd like to take a look at your protocols, so we can have agreement on those before you move forward. So justification for sample size derives from that non-inferiority margin, should be based on the targeted power, the non-inferiority margin, and reasonable assumptions of use success rates. And we've seen some folks looking at research articles and things like that to help determine what a reasonable assumption of a use success rate might be for their product. So assumptions of unreasonably high or low use success rate make the sample size smaller than what is actually required, and that would result in an underpowered study. And then the sample size itself should be calculated based on a method appropriate for paired binary data when a crossover study design is used. Okay, so binary data is use error versus use success. So some challenges that we recognize, we recommend conducting CUHF studies in subjects who are currently using the RLD, but we also understand that there may be challenges with recruiting enough subjects for that study. And so we'd encourage discussion with the FDA if you expect that that's going to be the case. And then for separate patient populations, for example, adult and pediatric patients, we may recommend separate subgroups to be statistically powered. Okay, so for considering randomization, again, we talked about the crossover study design, randomizing subjects of sequences between test reference and reference test. That should occur before the study is started. Systematic allocation of subjects to sequences can cause bias, so you can have sequence effect or some period effect. And that randomization scheme should be done using a proper statistical method. That may include, so we're to use your statistical methods to generate a randomization schedule, assigning each new subject to the next sequence in that randomization schedule. And if you have additional subjects that are enrolled for other than a regulatory purpose, you can use a separate randomization for those subjects. So if I want to have reference naive subjects, I may want to stratify my randomization between a reference experienced and a reference naive group. So some general recommendations about submission. Again, really would like for you to submit your protocol for our review and feedback on that study design so that we can agree on analysis methods on non-inferiority margin, before you start that study. And again, we recommend you consult your statistician or a consulting statistician for the statistical methods and sample size calculation randomization. General statement, online sample size calculators don't usually have appropriate methods for that calculation for paired binary endpoints. So tips for submitting your CUHF protocol include your threshold analysis. We need to clearly understand what the user interface design differences are so that we can then assess whether the study's been designed appropriately to address those. Make sure that you recruit appropriate expertise to inform your statistical analysis plan, explain and justify how you derived delta, how you derived your sample size. Provide 5 samples of your product. And then going back to that, the guidance, refer on the guidance, contents of the complete submission for threshold analyses. And then please wait before proceeding with your study so that we, again, can make sure that you have the right non-inferiority margin, the right sample size. Okay, and I think we're going to do questions on the panel. Thank you.
So while we're waiting for the slides to come up, my talk is going to focus on sharing some information about the research program that is GDUFA funded and what some of our goals are to try to address some of the uninformed or pain points that we know industry is experiencing. So this talk is generic drug device combination product research with a focus on comparative device user interface assessment and how differences impact users. So we know there are challenges with development of drug device combination products that relate to user interface differences. And that can get in the way of efficiently making generic versions of products accessible to the public. FDA-supported research about comparative user interface assessment and the role of published outcomes. We'll talk about that. We'll talk about the first study that has been completed in this space, which focuses on attitudes and perceptions of generic substitution of complex drug device combination products based on focus groups that were conducted. We'll talk about the current grant-based research that's ongoing, as well as plans for the future. So I mentioned known challenges. We know that controlling cost while avoiding violation of patent protections is challenging, and that choosing or developing a device user interface that's viewed as substitutable for the reference listed drug can be limited by these boundaries. If there are user interface differences that affect external critical design attributes, justifying those differences will not lead to a higher rate of user errors, will not alter that overall risk profile of the proposed test product compared to the reference listed drug, is a challenge. And how can you do that? What kinds of data and information are acceptable? Because we always talk about other types of information other than comparative use human factor studies, but when is it really possible to use other types of data, and what kinds of research can help make conducting and designing comparative use human factor studies easier? So, like industry, we're constantly trying to find where's the sweet spot. It is a balancing act all the time, trying to optimize access to affordable, high-quality generic drugs for the American public, while still ensuring that these approved products are going to be therapeutically equivalent between RLD and generic and substitutable at the pharmacy. We know that the majority of states in the United States allow generic substitution to occur at the pharmacy level without prior notification of patient or prescriber. We know that when a generic is substituted, that the user should not need any additional training beyond what they received when they were initially prescribed the product. And if there's a difference that may affect an external critical design attribute or adds a task, that data needs to support that there will be or is unlikely to be any increase in user error rates. But there are also some things we want to know that we don't know yet. Which types of task differences can users navigate without an increase in user risk? There's really no published data on this. The comparative use human factor study outcomes submitted to FDA are not published. We have not seen a lot of direct application of anthropometry data in this space. So it's been hard to leverage existing data to help inform this space, and we'd like to have more data. And then the perceptions and attitudes from patients are another piece of that patient-centric puzzle. Of how do people actually think through the process of using their products, and how are those mental processes and their prior experience and information they've heard from other people actually informing and affecting their actions. So here's our current goals. We have now completed the patient and caregiver attitudes towards and perceptions of generic drugs and generic substitution studies. So we'll talk about that a little bit today. We're going to talk about the goal of developing human factors-based tools and methods for helping industry and ourselves to better define and determine what is a minor design difference versus another design difference for particular types of products. And again, looking at what kinds of data can support and justify other design differences that are identified through comparative analyses. We're also hoping to conduct some studies that can actually get some data out into the published space. So the first study that was conducted with GDUFA research funds to try to look at how substitution of complex drug device combination products affects patients and how they perceive this was conducted through this contract that was awarded to RTI International and was just completed a couple of weeks ago. And the purpose was to advance FDA's understanding of patient and caregiver attitudes towards drug device combination product substitution and learn how patients and other users perceived and processed these design differences that were put in front of them, and to use this evidence as a way to help inform building policy and designing future research. This was a qualitative study that was done through focus groups, both in person and remote, because this study got caught in the middle by COVID. And they utilized a journey mapping exercise. There were two populations that used the same study design. The first one was a dry powder inhaler user population, and they recruited both asthmatics as well as patients with chronic obstructive pulmonary disease, both adults and adolescents, and the adolescents were limited to asthmatics. So they did not make an attempt to balance the population with an equal number of asthmatics and COPD patients, so they were not equally represented, but both types of patients were represented. The second study population was users of epinephrine autoinjectors. And that group of focus, that set of focus groups included adult and adolescent patients with severe allergies, as well as caregivers of these individuals who might also be using the product. There was segmented recruitment to ensure that there was an adequate number of both adult and adolescent patients for both of the study groups, as well as caregivers for the epinephrine autoinjector study. For the dry powder inhaler groups, all the focus groups were finished in person, but only half of the focus groups for the epinephrine autoinjectors were conducted in person. And then during COVID, all of the study documents were actually rewritten in order to conduct the remaining focus groups remotely using the Zoom platform. And all of the groups included a pre-group questionnaire that was sort of a follow-on to the screening process. And then they initially in the focus groups, discussion was generated just about participants' overall perceptions of generic drugs. What did using a generic drug mean to them? And that was followed by the journey mapping exercise that was basically a 3-step mental journey through a situation they might really be in. So let's talk about what that is. What is journey mapping? So this is a process that's used in market research to understand consumer perceptions and about how consumers make decisions. And this journey mapping process was adapted for this study to understand how patients navigate complex systems and health decisions. And so it's been used in other settings related to health as well as in this study. And the 3 parts of this journey for participants was first, the participant orders a refill for their prescription, either dry powder inhaler or epinephrine autoinjector, and they talk about that process. The second part of the journey mapping process was that the participant goes to the pharmacy and picks up their prescription, and they unexpectedly receive a generic version of the product that they've been using instead of the brand. And then the third part of the journey mapping exercise and the discussion that followed explored what does the participant do, what do they feel when they sit down to use this generic product for the first time. So for steps 2 and 3 of the journey mapping exercise, whether the focus group was in person or was remote, at that point, the participants had the opportunity to interact with both the name brand product and the generic product. So for the focus groups that were done in person, they had a couple of different samples of trainers or of emptied inhalers that were on the table and could be passed around. For the remote sessions, these devices were actually, the trainers were mailed to the patient's house in a sealed package that they were told not to open. And they basically opened it online in front of the moderator so they could see what was going on. And so these are the dry powder inhalers that were used. The comparison was between Advair Discus and Wixella InHub. And you can see here that there were 4 groups of adults, and almost 81% of them had asthma, 22% had COPD, and 69% had received some sort of formal training on their Advair discus. In the one group of adolescents that participated in a focus group for the dry powder inhalers, 50% had been trained on their device. And the second group of adolescents was canceled due to COVID. But the researchers felt that based on all of the data they collected when they were reviewing it, that they had really saturated all of their responses after the number of groups that were conducted, and they felt they had enough to do a thorough analysis. The citation for the publication of this part of the study is on this slide if you want to find that later. For the epinephrine autoinjector group, the comparison was between 2 trainers, the epinephrine the EpiPen trainer, which you see on the left, and then the Teva epinephrine autoinjector approved generic that you see on the right. And 8 focus groups were conducted, 4 in person, 4 remotely. There were 3 caregiver groups, 3 adult groups, and 2 adolescent groups. And the outcomes and reactions between the in-person and the remote groups were very similar. So the researchers decided in their analysis to combine their data from these groups. And the focus groups were conducted in the same manner as with the dry powder inhalers. And the differences with these products as compared to the dry powder inhalers, just to go back to what's your population, what's your setting of use, here you have an emergency use product. It is not a chronic daily use product, and it may be administered by the caregiver and not just by the patient. And so those were 2 differences for this particular product compared to the dry powder inhalers. So as the participants sat down to explore their epinephrine autoinjector, some of the things that they noticed that were different were the size of the autoinjector, how it felt in their hand. They noticed that the EpiPen has a carry case that you tip the autoinjector out of before you use it, and has no cap over the needle end, whereas the generic has no carry case, but has a twist-off cap over the end of the device. So I was hoping, I don't know if this has a little light or not. Does it? Nope, it does not. I think I just took myself out of presentation mode. Sorry. Can you put me back into presentation mode? I pushed a button thinking it was thanks, sorry. It's all right, I can get there. Apologies to everyone. So with these differences in design, the participants noticed that there was a different number of steps, and that raised some questions that they went on to discuss. So overall, the study results and takeaways were actually relatively similar for both the dry powder inhaler and epinephrine autoinjector groups. Patient and caregiver experience with and perceptions of generic drug products continues to expand and improve. Adolescents weren't quite sure what generic meant, but with some examples around things like cereals and that sort of thing, they got the idea, understood the concept, and were able to discuss it. And the perceptions as far as positive feelings about cost savings and positive feelings about the product was likely to be as good and work just as well as their brand name was much improved over what published data showed, say, 15 years ago. So the education of our public about the quality and reliability and substitutability of generic drugs is definitely improved. Patients and caregivers consistently appreciate the opportunity to save money, and that made a difference to them. But they had some lingering uncertainties, and they had anticipatory anxiety about would they be able to use the product correctly in an emergency? Would they remember to follow the right steps so that they could administer the epinephrine when somebody was having an anaphylactic reaction, whether it was their family member or themselves? So there were remaining concerns there, and it was really evident among the caregivers who really had a lot of anxiety around this. Patients were frustrated by generic substitution. not so much that it was being substituted, but that nobody told them it might happen and why. And so this is an opportunity, certainly not just for industry and for FDA, but for our prescribers to have conversations with their patients who are using complex drug products, complex combination products, about when they may receive a generic and that it could happen and that's okay, and to talk about it ahead of time. So additional plain language education for healthcare providers and patients about approval requirements for generic drugs may help alleviate some of remaining uncertainties about generic drugs. And then again, the idea of being prepared for what may happen rather than being surprised. So 2 years ago, we put out some requests for proposals for opportunities for grant funding. And I wanted to talk a little bit about the research that's ongoing right now with our grantee at University of Detroit Mercy School of Engineering. Our principal investigator is Megan Conrad, and what they are working on is developing a combination product taxonomy and comparative human factors testing method for drug device combination products submitted in an ANDA. And any of you who are in the room who get a chance at our next coffee break, I put a poster back on the table in the right back corner over there that you can take a look at. That is a larger representation of something I will show in a couple of slides that reflects some of the work that this group is doing. So the aims of their project, they started off with information gathering. They really wanted to understand the landscape. They conducted interviews with industry and academia experts in this comparative human factors area, or folks who have had experience doing it in their job. They conducted a literature review, as well as looking at various methodologies that have been used in the comparative human factors space. And they wanted to investigate different kinds of design attributes and whether there were any taxonomies or models that were already out there that they could build upon. So aim 2 was to develop an actual visual taxonomy with a goal of creating a library of existing drug device combination products and their user interfaces, and developing some sort of visual classification system that would help break down the various characteristics that make up a user interface for a device, to help make it easier to look at two different user interfaces and more readily see what's the same and what's different. And they wanted to develop a database to go along with this that would include any known information about potential use errors. And then aim 3, which has not been started yet, they're far along in aim 2, is to actually conduct comparative analyses using the taxonomy that they develop and validate, and conduct comparative use human factors studies or data collection, or explore different methodologies to then evaluate any identified other design differences. So from the interviews they conducted, the positive feedback they received was appreciation for the expectations outlined in the draft comparative analyses guidance, and the considerations for how to approach the process. But the folks they interviewed were frustrated by different objectives that were described in the FDA guidance for comparative analyses versus human factors validation studies. And there was just confusion and frustration about those differences, the process for calculating sample size, the complexities around that, because there's an inability to obtain RLD data, and because nobody's publishing their data. The lack of methodology for computing and categorizing errors, the failure to address severity of errors, and the confusion about calculating acceptable error rates. So these were some of the frustrations that just came out in the industry interviews. So taking that information, as well as thoroughly going back and reviewing the public comments that were submitted after publication of the draft guidance on comparative analyses, the research team decided that they wanted to identify all the points of user interaction with complex devices. And they decided to focus on pen injectors, prefilled syringes, auto injectors, metered dose inhalers, dry powder inhalers, and nasal sprays as their primary complex device types to look at. So first they started by saying, okay, let's look at different ways to categorize inhalers. And they started first by their sort of mechanistic categories. So they grabbed all the metered dose inhalers and put them together. They took the dry powder inhalers and divided them into multi-dose versus single dose, and then the soft mist inhalers. And so they showed this to us one way, and they said, okay, this is one way to do a visual classification system for inhalers, but we can also do it a totally different way. We can do it by shape. We have elbow designs and cylindrical designs and disc designs, et cetera. And so they were really sort of mentally exploring what makes products similar and what makes them different. So having gone through that exercise, they started developing a visual taxonomy related to user interface design differences and use-related risk. They wanted to find a way to both simplify and standardize the process of identifying minor versus other differences. So, and this relates to both physical design differences as well as task differences, because the task differences affect how people interact with their user interface. And so that brings us to the development of their visual taxonomy. And you can see here that their whole process or idea about linking the design features to risk marches through categorizing, looking at task analysis, looking at use error analysis, risk assessment, and then linking this to the user interface elements. And this whole process very much parallels the iterative design process that so many of you are going through every time you're developing a new drug device combination product. So this is only a piece of what they've started to develop, and you'll see the more thorough layout of this prototype back on that poster if you take a pic. And they started breaking down all of the different considerations, sources of information, things that the user actually has to think about and either receives in a sensory way from what the device is telling them, or things they have to do to the device. So they looked at different ways training materials are provided. They looked at different ways that instructions for use can be provided to the patient. They looked at on-device labeling, audible, visual, and haptic feedback. They looked at different kinds of controls. Does the device have push buttons? Does it have levers? Is there something you have to slide? Is there a touch screen involved? Et cetera. So they looked at all of the different potential controls that a device might have, and they looked at the interaction points. Where does the user have to hold it? Do they have to plug something in? Do they have to connect two pieces? How does that work? And then what are the characteristics of the main body of the device? And they took this breakdown of categories, and then they applied it to 3 different inhalers. So we don't expect you to be able to read this, but if you just pay attention to the different colored boxes on the different lines, you can see, looking across Advair Discus to one of the Ellipta devices to an MDI, looking across that row, where different aspects of this user interface for the device are similar to one another and where they appear to be really different. And this is their whole goal, is to try to create a very visual way, sort of a visual and tactile way of comparing all of these different aspects of a user interface. Let me back up for 1 second. And so this work is ongoing, and we're pretty excited about what they're doing. And we've even had preliminary discussions about if this, as it's piloted out to people and validated, if people find it useful and find that it would help their work process. Is this something that could even exist in an electronic way, where it becomes something that you can basically fill out and make selections and have a graphic display for your user interface comparison? So the next piece of research I just want to briefly touch on is that we have an IDIQ solicitation, stands for indefinite duration, indefinite quantity. It's a large contract under which multiple task orders or studies can be conducted. We hope to use this request for proposals opportunity, which just posted, I think about 10 days ago. We're hoping that we will get proposals that allow us to actually conduct some comparative use human factor studies so that we can compare user interface differences that we've seen as a challenge that we think industry is struggling with, so that we have an opportunity to explore statistical methodologies in this space and get some data out there and published that all of you can then lean on and use when you're designing your own studies. I'd like to thank the RTI contract research team, the University of Detroit Mercy Grant research team, all of my research team colleagues who have been participating from a variety of sub-offices within OGD and within CEDAR, as well as my leaders, Rob Leinberger, Markham Luke, Sam Ronnie, and Jesse Flora, who's been absolutely incredibly helpful through the whole process with our research as well as with this course. So thank you all. I'd like to welcome up my colleague from Beatrice.
Thank you, Catherine, for the introduction, and thank you for the opportunity to speak on behalf of industry to the other design difference topic that we're looking at this afternoon. My name's Claire McDiarmid, and I'm from Beatrice. Let's wait for my slides to come up. So as I say, I'm going to speak to you about the challenges in demonstrating acceptability of other design differences as identified through comparative analyses. This won't be the first time you've heard this afternoon or today, but the development of drug delivery systems for generic products is a balancing act. We hear as generic applicants that we must scientifically demonstrate that the product performs in the same manner as the innovator. However, we're faced with a number of challenges on how to do that and a number of restrictions for various different reasons that I'll speak to as we go through. Whilst, and I'm saying this slightly tongue-in-cheek, but whilst developing a generic drug product, and I speak as a chemist who spent quite a lot of time in the labs, it is in some ways a copy of a formulation, and the challenges for drug product development lie in bioequivalence and the demonstration thereof. It's very rare that a drug delivery system can be a copy. We're restricted by patent restrictions. We have other challenges as well. Reference products may not be aligned with our contemporaneous user needs. We've heard a number of people say today, what happens when the drug delivery system that's your reference product is old, maybe even not considered to be a combination product at the point of application? How do we navigate that challenge? The understanding of the risk of use error prompted by any differences that we might identify through comparative analysis is key to the development of the proposed generic and the acceptability of those differences. It's been great, actually, in the introductory speech from Dr. Leinberger this morning, we heard, I certainly picked up on the term, this is an innovation frontier. And that's really nice to hear, that as a generic industry, we have the opportunity still to be innovative, to work with the agency to develop process and methodology that will help us to bring accessible products to patients. It's not the first time that as a pharmaceutical industry or a device development industry, we've been innovative. We've seen through the years the development of drug delivery systems from the point in time where they were just considered plastic packaging and not even hardly mentioned in an application, through the development of human factors, assessment from traditional validation type studies through to comparative analyses and now comparative use human factors. We've seen that development, we've been part of it, and the agency have shared their thoughts and their contemporaneous approach through the issuance of draft and final guidance. And I think that's the key that I want to speak to today is where do we go next in this space? And one of the questions that I'd like to pose that hopefully we can speak to in the panel discussion later is, are we at risk of sacrificing enhancements in safety and product design by restricting allowable differences in the user interface? This won't be the first time you've seen this product, and you'll see it on another one of my slides, really using Advair Discus as an illustration of an innovator product that has led to challenges in generic design. And I can speak to that from a decade of working on generic Advair. Innovators, they're seeking to, in some ways, to block generic development of same and similar products. That can be through patent restrictions, and we've heard of that, and we'll suffer that forever, I'm sure, but also through marketing and branding aspects of an innovator product. These can speak to design, product association through shape, color, steps of use, and they can further restrict the generic user interface development space. What we're seeking to do as an industry and with the agency is to ensure accessibility and equity of generic combination products to the wider population. And the reason that we're all here today is to look at comparative analysis and comparative use and ensure that those methodologies enable the development of a comparable user experience. I'm going to flick actually to the next slide, because this is really just an illustrative slide of something that I'm going to speak to in a little bit more detail. A couple of examples, and both of which we've seen already today, of other design differences in the user interface. On the top left, we're seeing the dial out of a dosing mechanism compared to a spring-based button. This is something that's restricted by patent and can often be the sole other design difference in a pre-filled pen, which obviously requires further investigation in consideration of the risk profile of the product itself and the mechanism of action and the mechanism of the user interface. In the bottom right, you see again generic Advair, Wixella Hub, and Advair Discus. Very clearly, another design difference in this product is the orientation of use. The use of the name discus indicates to the patient that the orientation of use. The shape of the mouthpiece is a clear direction as well. And prior to the introduction of comparative analysis and comparative use, this was an area that I spent quite a lot of time discussing with the agency and with our developers as we brought this product to the market. So they're examples of the other design differences. And I've talked about patent restrictions. There are other ways that other design differences can arise. Mitigations against known use errors, and we've heard people speak to that already today. What if your reference product's been on the market for quite some time and market data, literature, patient advocacy fora are telling us that there are known use errors and something that we have the opportunity potentially in a generic to mitigate against. We also have changes in current clinical practice. and potentially the use of a platform design rather than a specific design targeted at a reference product. All of these can be reasons for the introduction of other design differences. So examples of that, I spoke about clinical practice. You may have an instructions for use for a reference product that directs the user to aspirate prior to injection. That's no longer a broad brush clinical practice. It's no longer used in all situations. So where do we put the balance for the generic IFU? Do we align with current clinical practice, or do we align with an older instructions for use? Another part of our balancing act. I've spoken of orientation for use and the touch button injector that you saw on the previous slide. I think the key thing that we're looking to work on here is that intention of the comparative use human factors guidance to confirm that other design differences that exist between the generic combination product test and the reference are acceptable, such that the test product can be substituted, the full expectation that it will produce the same clinical effect and safety profile. And that's something that I think we're all aligned in. But what I'd like to talk about is the challenges that we face in doing so. And I've been very pleased to hear, as the presentations have progressed today, that we're hearing from the agency that there really is an opportunity to use other methodology, that comparative use human factors is one methodology that can be used, but we need to talk about other options. And it may well be that there are good reason to look towards those other options in specific circumstances. So what are the challenges that we're facing? My purpose here is not to talk through the guidance, but to highlight elements that are causing us challenge. So we know that comparative analysis determines that we have an other than minor design difference, and that an assessment is required to confirm same and similar use. We know that there is a guidance on the use of comparative use human factors studies, the objective of which is to demonstrate that the use error rate associated with that change will not impact the risk profile of the product, and that we can test that through simulated use using participants representative of patients and caregivers, and that for each critical task that's impacted by the change in a critical design attribute, the use error rate of the test product is no greater than that of the reference product plus a delta. We've heard about paired and parallel design. So the draft guidance says that in some instances, it might be appropriate to focus the selection of user tasks on those critical tasks that are related to the critical design attributes found to be different. Herein lies our challenge. When is that acceptable? How do we justify that? Another consideration that I'd like us to tease out further in the panel discussion is, what role does root cause analysis play in comparative use study reporting? We all know that root cause analysis is key to the traditional human factors study reporting. But how does that play out in this situation? And thinking again to comparative use and the binary outcome that determines the use error rate, how does that compare to the identification of minor differences in a threshold analysis? So when you submit a comparative analysis with a bunch of minor difference classifications and one other design difference that you then go on to assess in comparative use, what happens to the data on the minor design differences, if indeed you have to test those out? And are we putting products with one or two other design differences? Are we setting those up to a higher bar than products with only minor design classifications? We heard Jason speak just before on recruitment. This is certainly an issue that we can face in any human factors study. Accessing patients, end users, whoever they may be, can be a challenge, especially when we're faced with products that may not be high volume. They may be fairly new to the market. We may have compromised user groups or limited access to HCPs. All of these will contribute to recruitment issues. And on top of that, the sample size determination in comparative use is in general higher, leading to higher participant numbers and potentially varying participant numbers as well to that of traditional human factors. So what can we do? What options do we have in study design to minimize that recruitment burden? We can look at the opportunities to assess high-risk users only. That is outlined in the guidance as an option. We can potentially look to recruit surrogate users where access to specific reference product patients is very limited. Again, that has to be discussed and agreed with the agency through protocol review. And then finally, whilst we've heard about paired design, and the guidance does suggest that paired design is the most efficient approach, could there also be an option for paired design where one of the arms is theoretical, in which I mean you assign an assumed error rate for the reference product as 0? This has an opportunity to reduce not necessarily your participant numbers, but other resources in terms of time, in terms of product, and will overcome accessibility issues with regards to the reference product, again, enabling us to progress through to the approval of a generic product as efficiently as possible. Potential risks to recruitment, and we heard this in an earlier presentation, could be in the determination of the reference error rate and the justifiable delta, in which I mean there is a possibility that we may have to introduce a formative study requirement or further assessment in order to identify those figures before we even start out on our comparative use protocol. Again, potentially leading to us accessing patient populations before we've even started recruiting for the study, and therefore limiting our recruitment even further. I've spoken to this already, so I shall move on. Arguably, the most challenging aspect of comparative use to date has been how to determine acceptable deviance to the reference error rate. We've spoken before about the draft guidance. I'm not going to talk right the way through this slide, but clearly what we're looking to do in a protocol for comparative use is to set forth our justification for an acceptable understanding of error rates and deviance in order to set up a non-inferiority test that will give us that robust justification for our other design difference. So how do we determine that delta? The guidance tells us that we can use the outcome of the threshold analysis to identify our other differences, and that there is anticipated variability in the error rate for the reference product. And there's no surprise there, because we all know, working in human factors, that you find some really random outcomes when you test in patients. We also need to consider the risk in any difference in the outcomes. The justification of the allowable margin that we need to present to the agency is somewhat less clear. What the guidance doesn't tell us is how to determine variability. What is the acceptable justification, and how to correlate the risk posed with the allowable margin? So I guess one request that I would be making on behalf of industry is that we can do something together that would enable us to provide worked examples and an indication of acceptable justification in the final guidance. And I think we've heard here from Katharine, and we've heard from earlier panel discussions that at the minute we're treading on new territory. We need to work on case-by-case basis, and we need to hear from the industry as the thinking it develops. But what I'd like to think is that we can get to a point in time where some of the low-hanging fruit can be set as an acceptance, as an example. Some of the work that Katharine spoke about there could be really beneficial to us as we look to further guidance. So in summary, we are facing challenges. It is a new methodology. It's inevitable. The clarity on study design requirements, that question of binary measurement versus root cause analysis and how the two can work together. Further guidance is required on the determination of that delta and how that fits with the risk profile of the therapy. We've had discussion on other design differences that could be assessed for an alternative route. How do we move that forward, and is there a framework that we can work within there? We talked about the possible use of early formative data as potentially guiding some of those alternate discussions. I've spoken of sample sizes, or access to specific user groups, and potentially overcoming this through surrogate user groups. Finally, the impact of protocol review timelines on the overall development program and this iterative discussion that we need to have with the agency. Whilst it's welcomed by industry, it would be really beneficial if we can slim that down somewhat through final guidance, providing further direction and ensuring that we spend our time talking about the key scientific elements of the protocol as we move into further understanding of other design differences. So questions we can take at the panel discussion. I'd just like to say thank you for your time and your attention.
So I'd like to invite all the panelists up for session 3. We've gone from running ahead of time in the morning to behind time now. So as the coffee break supplies come out for the 2:30 coffee break, please feel free to get up and stretch and grab a cup of coffee and a snack. If there are a lot of questions, we'll just keep going with the questions and the discussion through the coffee break. The other thing I've noticed is it's hard to know who's who. Everybody's on a list. But I would love to just, once everybody's seated, just go down the row, have everybody just say their name and their association so that you know who's who on the panel. Okay. So why don't you go ahead and start, Nitesh, and just say your name and who you're affiliated with and what you do, and let's work our way down the road just so as we're fielding questions, everyone knows who you are.
Hello, everyone. Myself, Nitesh Patel, Senior Manager from Sun Pharmaceutical Industries Limited. I have been associated with Sun Pharma since more than 15 years and responsible for quality submissions of ANDAs and 505(b)(2)s. Yeah. and also responsible for review of human factor studies and formative and validations protocols for studies. Yeah. Thank you.
Claire McDiarmid, we've met.
Somesh Chattopadhyay. I'm a team leader in the Division of Biometrics 8 in the Office of Biostatistics in the Center for Drug Evaluation and Research at FDA. And I have been actually involved with the review of comparative use human factor studies for last at least 4 years.
Jason Flint, I'm the Associate Director for Human Factors in DMAPA 1, Division of Medication Error Prevention Analysis 1, responsible for reviewing use-related risk analyses, comparative use human factors, HF validation, et cetera.
Hi, folks. Irene Chan, I'm Deputy Director in the Office of Medication Error Prevention Risk Management. So our office has oversight basically at looking at labels, labeling, proprietary names, basically anything that relates to use errors, medication errors that can occur, and also the oversight of risk evaluation and mitigation strategies or REMS. So the human factors work also falls under the Office of Medication Error Prevention and Risk Management. Thanks.
My name is Chong. I'm the Director for the Office of Safety and Clinical Evaluation. Our office is slightly less of a mouthful than Irene. We oversee the clinical assessments of ANDAs. We've heard from several people from our office in the Division of Clinical Review earlier, and we participate in the review of comparative analyses for generic drugs.
Hi, Markham. I've been introduced before in the Division of Therapeutic Performance in the Office of Research and Standards. The Office of Research Standards does a lot of work in the pre-ANDA space. We help guide generic products as they develop their products before they submit the ANDAs. Thank you.
Thank you all. I'd like to go ahead and see if there are questions to start with in the room. And if people are still thinking about them, we'll see what's online. Any questions in the room right now? There's one right here in the middle.
Thank you. I have a question for Dr. Jason Flint. In your presentation, you said that we have to have a separate randomization for patient subjects and for naive subjects. So even if we include a provision of the naive subject in the protocol to begin with, still we have to have a separate randomization for both kind of subjects?
I'm going to aim that at my statistical colleagues.
Okay. Can you?
Sure.
I mean, can you repeat it once more, because I didn't get what you meant by two types of subjects there.
One is the patients which we enroll, and other is the naive subjects.
Oh, naive. Okay.
So in the one of the slides, it was mentioned to have the separate randomization for both kind of subjects.
Yeah, that was an example.
Mm-hmm.
So if naive subjects are not acceptable for the study, then use a separate randomization for that. That is the recommendation.
Okay.
That is a general recommendation for any statistical, any study, any clinical study. If you are randomizing, just separately randomize those who are going to be evaluated in the study than those who are not going to be evaluated in the study for the regulatory purpose, because otherwise the randomization will be mixed up. So that is a problem.
Understood.
There's a thought there that the patients who are not naive, had prior experience with other devices, would have a bias of some sort.
They may be different, so we don't know that. That's why we want to have a separate randomization in that case. And it is not difficult to do that.
Okay. I have a follow-up question here. If we start our protocol only with the patient subjects mentioned there, and later on we feel there's a challenge in enrolling those patients for this study, can in between we amend our protocol and include the naive subjects for this study?
You want to take it?
I think you would want to come back to the agency and make sure that you're recruiting. Like, is this you have to be sort of particular about, like, have we already had a discussion about who the surrogates might be and right, and whether we already have agreement on that, right, to make sure that you're recruiting sort of the right folks. I don't know if you had anything from the stats side. Did I understand your question correctly?
Yeah. So means your recommendation is that we reach out to agency and take their concurrence before we include the knife subjects in the study.
I think that there may be cases, and I sort of alluded to that when you're building your CUHF protocol. If you foresee that there are going to be issues, it may be something you want to address in your protocol, right, ahead of time. So if we already have agreement on that, then it sort of makes that discussion a little bit easier.
Okay.
Yeah.
And if in between, if we have to come back to reach out to agency, will it be through email or through control correspondence?
Catherine, do you have an answer for that? I don't.
Can you say this again, please?
Means like the recommendation was, if we have to include the knife subjects in between of the study, we should reach out to the agency to take the concurrence from the agency. So what?
This is I would say probably not by email. I think using one of the formal meeting pathways would probably be best, either control.
Yep.
I don't know if this would qualify under pre-ANDA, but certainly email is probably not the best way.
The reason I asked email, because control correspondence will take 60 to 120 days and studies in between. So that's why I'm asking, what will the fastest way to get the feedback?
Yeah, I understand that can be a challenge. I think I would reference back to Jason's earlier comment. If you have beforehand consideration that this might be a problem, if you build that in the protocol up front, that if we, other surrogate populations that we might want to study, if we're unable to recruit, that might be a way that you don't have to come back, but it does require a little bit of forethought in terms of what you might encounter in the future. There's a little crystal ball reading, which is always difficult to do.
I mean, we understand that the time is an issue, but at the same time, the quality of the advice is an issue too. And so those review time periods are to make sure that we understand what you're asking and can provide as informed a response as possible. Betsy, is there a question online that you would like to pose while we're bringing the microphone over here to this nice lady who has a question over here?
So there's several questions, mostly regarding how determining the non-inferiority margin. So, Somesh?
Yeah, this is a joint effort with. It is not just a statistical issue. It is an issue that the determination actually involves all disciplines. So everyone has to be involved in that. How the determination is done. Usually for other studies, you may have seen actually the non-inferiority guidance for drugs and biologics that has some methods that is mentioned there. But you, in this case, understand that for devices, it may be a little difficult sometimes, especially the effect, because you're not determining the efficacy in this case, but how much error you can accept. That is how much more error, how much you are sacrificing from the RLD. That is the question here. So the non-inferiority margin will actually determine that. So the non-inferiority margin is not based on the sample size. The sample size is calculated based on the non-inferiority margin. So the non-inferiority margin is how much less success we can accept. And that is not just a statistical, there's no statistical answer to that. But determination actually involves, of course, past literature, and what you have seen that also it was discussed in today many times, that there is not enough data outside. So that is a challenge, of course. So one possibility is just to conduct some small sample study, like formative study. Yeah, formative study or some kind of study to get some estimate. So that is one possibility. But if there is data outside, then of course that is the best way to get some idea.
And Markham, you had something to add?
Yeah, Somesh and I've chatted about this as well, that when you don't know what your sample size might be, but you have some estimates or guesstimates about that, there are approaches that FDA as both CEDAR, CDH, we're all trying to adapt adaptive design to our studies. And then the other thing in lieu of a formative study, you could do a study as a pilot study and declare some of those as priors via Bayesian approach. And so we're open to alternative statistical approaches to study design to meet the challenges in this space. I think it's something that is relatively new ground, but it's been done before in other centers in FDA, and there's a lot of literature on how to do that appropriately. Thank you.
So something that, thank you.
Sorry about that.
Something that, I've mentioned before in other settings. Remember, at the end of the day, you've heard this before in the presentations. What we're trying to establish is that you have a product that's therapeutically equivalent, right? Which means, and I'm not quoting because I can't remember the exact language in the statute, but at the end of the day, you're trying to look that to ensure that you're providing a generic product that effectively has the same overall safety and efficacy profile. And so when you think about it in terms of that lens, I think it's important to think about when you're thinking about the margin, you're thinking about what difference effectively am I going to allow in this test that won't put me past the point at which I'd be looking at a product that would have a different safety and efficacy profile. And I think that's important to be thinking about, is what we're trying to reach for, what we're aiming for here. And so that would be part of your justification as you're thinking through what is important as a consideration when I say, I'm justifying this because this amount of difference is not going to be clinically significant. That's something that you can also be thinking about. But it's important to also do your due diligence and take a look at what is available in the literature, what is available out there to give you direction as to what is known and seen in terms of errors with, for example, the reference listed drug. So just a couple of things to think about.
And just one more. I know it's a popular topic, but to Somesh's point, it's not just a statistical consideration, right? That you have to consider that what's the difference or what's sort of the harm associated with the error that may occur, and both in terms of harm and in terms of efficacy, right? And so that has to play into the equation. That may be a very different number. You may have a very different non-inferiority margin for an emergency use product than you would for a non-emergency use product, or for a product that has a narrow therapeutic margin versus one that doesn't, right? So it needs to be very clear. It's not just a statistical consideration.
And there's a question on front over here and another one over here next.
Yeah, my question was actually, Markham partially, he just responded. Why not think about an adaptive design, like where we do an interim analysis and then define the non-inferiority margin?
Yeah, it is possible. So actually, we are open to new designs, but you have to, actually the industry has to submit the protocol for any adaptive design, that it has to be, actually you have to review this and then you have to decide whether that is acceptable. So that is possible, definitely.
And then you can also do the simulations, right? Using the Bayesian stats. If you do a small sample size and then you...
Yeah. So we don't have any objection to any methods. The only thing is that you have to submit your plan and make sure that you have the details so that you can evaluate them.
Yeah, thanks.
I think there's a question back there with Brandon, and we also have another question. Anna, there's another one right here. Okay, so why don't we go ahead and start with Brandon? Thank you.
Sure. So I guess first, thank you to the panel for taking the questions. But this is, I guess, directed to Jason and Somesh. So in Jason's presentation, it said if there was disagreement on the non-inferiority margin, you may have to repeat your study. So sometimes even after corresponding with the agency, there could be residual uncertainties in what the non-inferiority margin should be. So one way that we've tried to combat that is to increase and overpower the study from a sample size perspective, so that even if there's disagreement on the non-inferiority margin, if it's tighter, we'd still have adequate statistical power in the number of the sample size that was employed. In such a case, is that an example of where you would still expect the study to be repeated, or is it, again, case by case?
Usually, if the non-inferiority margin, I mean, even if we do not agree to the non-inferiority margin the applicant sets, but if we see that the study has shown non-inferiority with respect to something that we might have accepted, then of course you don't have to repeat the study. But then still you have the uncertainty that you don't know what you'd accept, because we have to actually check. We have to do some research there. So it is better that you do the research and set the margin first, and then conduct the study.
Hello. My question is related to one of the slides which Claire has presented regarding two products, Victoza and Saxenda, where both products are from the same innovator company, but both devices are having a different technology. And more or less, the one is for diabetes, one is for obesity, but many times the patient population is also common for both devices. So in generic, when they develop, mostly the generics are having a single device platform, and one of the device will differ from the innovator, while the same device is getting used by the innovator for other products. So what are the agency's views? Because that definitely will turn out as other design differences. But will it really need a human factor study, or is there any other way out based on the same company innovator product? Is there any other way out to justify those things?
I think for the benefit of the whole panel, just want to make sure we understand. So you're talking about a scenario where there's an innovator company that markets more than one presentation of a particular active ingredient. So there might be one that's marketed in presentation A, another that's marketed in presentation B, with differing indications associated with each of those. Is that the scenario you're talking about?
Yes.
And then at some later time, a generic company is coming in. And I think the important question I would want to understand is what indications being pursued by the generic company?
For both.
Okay. And so and this is a hypothetical, but obviously it depends on labeling specifics and some other considerations here. But let's say hypothetically that the innovator company has one application, one label tied to one design and one indication, and similarly a separate application, separate presentation tied to another one. I think it's important for that generic company to be clear on which is the RLD and what are they pursuing in those instances. And it might be that you're pursuing two generics, right? Two generics, one for each RLD.
Yeah.
And so in that case, I think, again, these are hypotheticals, right? So you could see how very quickly it depends on the particular case in front of us. But if we follow along with this logic in this scenario, I think then what you'd be looking at is coming in. And if your decision in that case is you actually, unlike the innovator company, only want to produce one platform, and let's assume one of those platforms do in fact align with the design of one of those two originator applications, making a lot of assumptions here, folks. then yes, I agree with you. That would probably indicate the other, for the other application or the other product you're pursuing, there may be some differences. Then the same thinking though applies. You still need to work through what are the differences, understand what they are, how they may or may not impact critical tasks. And so the agency would still be looking for that thinking and analysis to have been done and submitted to the agency for further input and discussion on the right path to follow. And I invite others on the
wanted
panel
to add to
on.
that briefly. So if you were trying to justify basically developing and using the same device user interface for products that would reference two different reference listed drugs, you would need to conduct two different comparative analyses against each of those reference listed drugs. And the types of differences in user interface that might bubble out of those two comparative analyses would not necessarily be the same, even though you're dealing with very similar patient populations, different indications, but similar risk factors. And what you would need to do to support those differences would really be driven by the outcomes of those comparative analyses. We, I think, as a group of folks within the agency who work together all the time, asking ourselves a lot of questions about this, often ask, as more comparative use human factors data or other kinds of data that can justify and support a particular type of difference between, two different user interfaces. One of the questions we ask is, well, if we have data coming in that show that a certain patient population can navigate this difference without an increased risk of user errors, when do we have enough data? And what is our ability to use data that we've learned from one application to help inform decisions about another application? So number one is we don't necessarily have data that justifies yet that the user interface differences that you're talking about that might exist for very similar products, but for different indications, we don't necessarily have data in hand that can even tell us whether there's an issue with increased user errors. And until we have that, we still need the data. And then even once we have the data, there are still some questions about how we can reasonably use that data to inform our decision-making. So I would say until we get further down the road in this drug device combination product space, you have to approach each of those comparisons individually, identify your minor differences, identify your other differences, and think about what kinds of data do I have access to that might be able to justify this, or what kinds of data might I need to generate to support it. We're still early in this space, so that's sort of where we are.
Yeah, I would just add that, yes, there's lots of assumptions built into sort of the conversation around the example. I do think that it's important, and we've heard this multiple times, that another difference does not mean that you must do a comparative use human factor study, depending on how your platform compares to product A and product B, depending on which product is different, what the other difference is. It may be that something can be supported without a comparative use human factor study, and that's something that you could propose some justification for us to consider and work through. And I think Katharine highlights a very good point that we're all in this sort of new innovative space together, trying to figure out what kinds of data we need, what we've seen, kind of what information we need from you, what information we can get it from research. So there's I think a lot still to be developed, still more to come. And I appreciate it's not a satisfying answer to say, well, we don't know yet. But it's kind of the answer right now. Some of these things we don't know yet.
And I'd like to ask Claire to share a little bit of high-level information about a creative approach that her team took to an approval that I think demonstrates that if you get creative and if you're product fits the bill, that you can provide very useful, informative, justifying data that's not from a comparative use human factor study. So please, Claire.
Okay, can I, yes, you can hear me. So Catherine's alluding to the generic advert development that I spoke to in my presentation. And I guess I should just caveat that this work took place before comparative analysis, comparative use guidance was available. But you can see how this type of assessment could still be valuable today when we're hearing invitations to speak to other methodologies. And the other design difference that we were investigating was the orientation of use of the generic user interface in comparison to the innovator. We had, so firstly, what I would say is, considering the risk profile of this product, we may have identified this not to be a critical task, or we may not have considered the risk profile to be high in this situation. However, in other dry powder inhalers, this could have been a very different scenario. So that speaks again to thinking about risk profile early on when you're setting your assessment. But in this individual case, our concern was orientation of use. We first set out to collect some lab data on the impact of orientation of use on the delivered dose and the profile of that dose delivery. With that data in mind, we then sought to test simulated use with innovator patients and understand that ability for them to switch intuitively from the horizontal orientation to the vertical orientation of inhalation and back again. And we built a protocol that looked at that crossover between the different orientations of use. And we did not in any way set out to test every orientation that was possible. And if you imagine, there are 4 orientations that you can put an inhaler in your mouth. That formative, if you like, that subjective data that we brought back from that study, whilst we had numbers, we knew how many participants had used the device as we intended it to be used. We also aligned that with a deep root cause analysis that helped us to understand what it was, if anything, about the design of that device that drove the use that we observed in simulation. We then aligned that with the data that we had that confirmed that at any orientation of use, the dose would be adequately delivered, as long as the participant was able to produce the inhalation profile that is anticipated. And so really what we're saying is that it was not in any way a statistically powered paired design, but we managed to gather the information that we could set alongside laboratory data and present to the agency in such a way that we were able to really demonstrate that A, we had an intuitive intended use, and B, if that were not to be the case and a participant did hold the device in a different way, that we had a really good rationale for that. And it was not a rationale that we could mitigate against through design warnings or labeling. And I think that really sets a scene for potential other alternatives. But that was not done in isolation. We navigated that protocol in detail with the agency before we set out to test.
Thank you so much. Go ahead, Markham.
I just want to say kudos on that because it set the groundwork for expectations for the American public as far as what to expect for a generic drug device combination product, and that it doesn't have to be exactly the same. Like just because the reference pill is purple doesn't mean that the generic pill has to be purple also.
I want to give everybody a chance for 5 minutes to stretch their legs, do whatever else they need to do. I wish we had more time to continue this Q&A. Was there anybody else with a hot burning question on the floor here before we go to break? Okay. Thank you all so much. Really appreciate the active discussion.